Hello and welcome back, friends. Today I'm going to explain how to use Translate tool from ExPASY and how to find the correct ORF with an example demonstration. Please like and subscribe to my channel to motivate me to create more interesting videos and suggest me a topic which you would like to understand through my video. Let's begin with ExPASY, which is a resource portal of Swiss Bioinformatics Institute. This portal and its tools are free and easily accessible online. Translate is one of the tools which allows the translation of DNA or RNA sequence to a protein sequence which is made up of amino acids. Why do we need to translate a nucleotide sequence into its corresponding protein sequence? It helps us to identify a correct open reading frame or ORF. A correct ORF is a stretch of DNA or RNA code that begins with a start codon, which is ATG, ends with a stop codon, could be TAA, TAG, or TGA. If you're using an RNA sequence, then the start codon will be AUG and stop codon will be UAA, UAG, or UGA. In this video, I'll be first demonstrating how do you choose a nucleotide sequence using the nucleotide database. Then we will use it in ExPASY Translate tool to get ORF, and we will learn some features and how to select the correct ORF. So let's start with obtaining a nucleotide sequence from the NCBI Nucleotide database. If you want to learn about this database and the sequence retrieval process, please watch my earlier video, for which the link is in the description box. Here is the process of retrieving the nucleotide sequence of human myosin light chain gene, which I'm using just an example. We choose the mRNA sequence for this gene, which will be translated in the translated tool. First, we have to get the sequence in FASTA format to use. Let us open ExPASY Translate tool from its website and paste our FASTA format sequence. On the right hand side, we have quite a few translation options. By default is the compact output. At the bottom, you can see that both forward and reverse strands are ticked, which indicate 5' to 3' and 3' to 5' directions, and will later be translated in 3 open reading frames each. Let's first see the verbose option, where the start codon methionine is written as MET or MET, and there are spaces between residues or amino acids. Amino acids are written as single letter codes. The default compact option is more convenient to visualize, as methionine is indicated by a single letter code M and the stop codon as a gap with a dash sign. Also, there are no spaces between the residues, making it really compact. Next two options also include the given nucleotide sequence. As you can see, the codons corresponding to its amino acid. There are spaces or without spaces as seen here. Now let's check the key features. I'm showing here the compact output. You will notice that there are 6 options for OR frames provided, namely 5' to 3' frame 1, 2 and 3, and also 3' to 5' frames 1, 2 and 3. The 5' to 3' options are from the forward strand, and 3' to 5' options are from the reverse strand of the sequence we had input. You can notice that residues are in single letter code and some of them are highlighted in red. The letter M indicates start codon methionine. Also, we notice gaps in between which indicate a stop codon or the end of translation. All the red highlighted parts or stretches show the longest possible stretch or open reading frames which begin with the methionine and end with a stop. But why there are so many ORFs? Translation can begin with any methionine, but if a stop codon comes too early after the start, it intervenes the translation frame, making the translated sequence non-functional. Therefore, we will be looking for a red highlighted ORF where there are more than 100 residues at least, without any stop codon in between. With a careful look at all the 6 frames, we can make out that the longest uninterrupted stretch is present in the 5' to 3' frame 2, which is for 194 amino acids, which means that this is most likely the correct ORF, which actually gets translated in a live cell, while other options are smaller stretches are not likely to get translated. We can confirm our results by downloading this ORF and performing a BLAST-P analysis of this ORF to see if it matches with other homologous myosin light chain proteins in other databases with more than 90% identity. To download only one ORF, you take the cursor to the M residue of the frame and click it. It will show one pseudo entry in its FASTA format. Pseudo entry is a temporary ID and a virtual sequence which is not the part of any database. Copy the FASTA format sequence to use in BLAST-P. You can take help from my video on BLAST-P by using the link in the description box. I pasted the virtual sequence in FASTA format in BLAST-P window, and here are the matching results which I have filtered for human sequences only. The two results show 100% and 96% identity, proving that this ORF is the correct ORF. Thanks for watching this tutorial on Translate tool to find ORF. Hope you understood how to use this tool and select the correct ORF. Try practicing by finding the correct ORF with your own choice of sequence and verify it with BLAST-P. If you want more details on ORF, please check my next video. Thank you, do take care and see you soon.